So my name is Abraham Growcott, and I work for Biosecurity New Zealand in aquatic surveillance. I've been at Biosecurity New Zealand as of yesterday for 13 years. So yeah, that's pretty wild. Started out in risk assessment and moved over about 8 years ago to aquatic surveillance. Today, yeah, thank you for coming. Today we're going to talk about how we can help detect species in the aquatic environment a lot earlier, so that we can have a few more options around management. The focus is going to be on marine biosecurity mainly, but we've got a little bit of freshwater in there too. So let's get into it. So the key topics for our session today will be some good old background, followed by the nitty-gritty about marine pests that are not in New Zealand presently, and marine pests that are in some areas of New Zealand, but we're attempting to halt their spread. We're going to talk about how to report potential detections, where to find info if you want to dive a bit deeper, and then resources. In the chat there is 2 links to a marine and freshwater pest ID guide. This presentation's mainly based around the marine pest ID guide, so feel free to click on that link and follow through this presentation once we get to the how to ID species section. So this is the good old biosecurity system diagram that a lot of us show in our presentations. I quite like it because it's a nice representation of the biosecurity system, and it's represented as a series of nets. So these nets represent an intervention within the biosecurity system, and we start from offshore. Sorry, I'm just getting my mouse. We start from offshore here, which is on the left side, and this is what the interventions are that happen before goods or people or vessels come into the country. And we've got these little red kind of, let's call them fish. These fish are biosecurity risks that potentially could enter into the country. So each net catches a portion of these biosecurity risks. Ideally, the first net in this diagram would be a bit bigger, because this offshore net is our best protection. It catches the most biosecurity risks for the least cost. That's because it's preventative. If you're coming into New Zealand and your boat is free of biofouling, you've prevented those organisms entering the country, and it's the easiest way. Compliance with our rules is the easiest way to prevent organisms coming to the country. But as we know, not each of these interventions are not 100% effective. So we have follow-up nets to catch any of those fish or biosecurity risks that slip through. So the next net is the border intervention, and that's the one that the public are mostly au fait with. Watching border control on TV. That's the quarantine officers doing inspections. That's when you come through on an international flight, the quarantine officer with the sniffer dog finding the apple that you forgot that's in your baggage. That's the border intervention. So making sure those rules are complied with. Today, we're going to focus on the next net, and that is behind the border. So that's surveillance and incursion investigation. So that's for any organisms that slip through the border and potentially establish in the environment. So this is where we can have a big impact by working together to champion early detection, because we all have a role to play in this. If we all work together, we can detect things early and manage them much better. A very good example, non-marine, is the yellow-legged hornet up in Auckland at the moment. So the public is a vital cog in that response for detecting nests and individuals. So this is where we're going to play today. But then we have these follow-up nets, which is readiness, response, and pest management. So if we can't eradicate the species in the incursion investigation stage, maybe the species is in multiple locations or at high densities that can't be managed, then we move on to response where we can surge resources to try and manage that species. And then finally, pest management if it can't be eradicated. In the short to medium term, it moves into pest management programs, and that's where a lot of our regional partners, regional councils, iwi have a role to play in that regional management. So this is our team. Manager is Mike Taylor. So if you've ever rung the 0800 Pest and Disease hotline, you're probably engaged with maybe one or two or more of the members of the team I'm part of. So yeah, we're incursion investigators and surveillance staff members. So just very briefly, what are the main pathways of entry for marine organisms into the country? Probably comes as no surprise that international and recreational vessels are major pathways. So we have ballast water as a vector within that pathway, which is mainly related to commercial vessels. And then we have biofouling, which is any vessel can have biofouling on its hull of exotic organisms that are not present here. Other pathways, not so important into New Zealand is aquaculture, but it's definitely a major pathway of spread overseas for organisms between countries. And then we also have the aquarium, ornamental fish trade. For the marine pests that are present in New Zealand, there was some science, a report done a number of years ago that looked at the species that were present in New Zealand, non-Indigenous species, and tried to figure out what pathway or vector they came in with. And it was assessed that the majority could have entered into the country via biofouling. So biofouling is a really big one. All right, let's get into the nitty gritty. So if you've got your Marine Pest ID link open, you can follow through. The next slides will have a page number, and that's the page you can go to look at the organism we're discussing. So for all of these pests in this section, they are not known to be present in New Zealand. So we've got that little map in New Zealand on the top right there with that line through it. So these are not known to be present in New Zealand, but we're on the lookout for them, and our surveillance systems are designed to detect them. So the first one is aquarium caulerpa. So this is Caulerpa taxifolia. So this is not exotic caulerpa, which is present in New Zealand. We'll get to that those species later. This is aquarium caulerpa. So this is on page 8 if you're following through with the Marine Pest ID link. So most likely for this species, it's most likely to enter New Zealand and the environment via aquarium release from someone's aquarium. So it's very bright green. It's almost like an unnaturally bright green as a diagnostic feature. It has a midrib, which is number 2 there, which is the stem, and then it has these little branched leaves that grow at the same spot on both the left and right sides. So these little leaflets and that midrib are the key diagnostic features. As you can see there, can be up to 15 centimeters high. The whole structure, and then there's these horizontal runners that the leaves are connected to that are in the sediment. So this species can form as like for other caulerpa species, dense meadows that cover the seafloor and rocky reefs. So potentially preventing other organisms existing in those habitats. This species also, when it's grazed by fish and other species, can produce a toxic compound to prevent that grazing, just like a lot of terrestrial vegetation. When it's grazed, it releases chemicals to prevent that. Um, it can grow on kind of sandy, muddy sea floor, and it really likes what is it? What is it like? It really likes sheltered environments. So bays and harbors will be the key area where it reaches that high density. Not so much the open ocean. So that's
Hey, did
so.
you see a question in the Q&A?
Oh,
Someone's just asked, is Calliope in aquariums in New Zealand?
there have been detections previously, and they've been dealt with. So no, it's not permitted for importation in the import health standard.
Thank you.
No worries. Yeah, just sing out if there's any other questions, and please ask. It's good. Awesome. Moving on, we've got the Chinese midden crab. So this is yeah, its name's pretty good. The key diagnostic feature is these hairy mittens. Um, this species has distinctive notches between its eyes, so two of them, and then on the side of its shell, on its carapace, it has four spines. As you can see, it doesn't have paddles at the back, like our native paddle crab. So using all of those diagnostic features, it's a pretty distinctive crab. Those middens are, yeah, no other species in New Zealand of crab has that kind of fur. This species is really interesting because it lives in freshwater environments too. So as an adult, it migrates up into rivers. And in Europe, it's got to super high densities in some rivers, like the Rhine River in Germany, and it'll create these burrows in the banks of the rivers, and then that can cause a lot of erosion because they get to such high densities that there's all these burrows or holes in the riverbanks that they just start collapsing, and that causes erosion and follow-on problems. So what this species does is it then migrates down to estuaries and the sea to reproduce. So it's quite unique in that aspect. Um, yeah, it can like a lot of marine pests and non-indigenous species, they can form very high densities. And they're quite generalists in their diet, so they eat whatever they can get their claws on. So they can, yeah, reduce native species, biomass or numbers quite a lot. And they can survive quite well in polluted waters. So, a lot of our ports and that are quite highly modified. So they can do quite well if they do get here. So there's a lot of available habitat. So this species could come into New Zealand most likely either as biofouling, as a juvenile or adult, or via ballast water as larvae. The Northern Pacific sea star. So this is one we're on the lookout for. This species is present in Australia, so it's actually not far away. Um, it's quite a big sea star. It can get to dinner plate size. Key diagnostic features is that it has 5 arms, and at the end of each arms, they're pointed and they're often upturned, so they turn up. Um, The color can be quite variable, so color's probably not the best diagnostic criteria for this species. Um, but they also have these spines on their top surface, which are randomly um, placed on the top, whereas a lot of other sea stars have rows of spines. This species does not. So you've got your 5 arms, they're pointed, they're often upturned, and you've got irregularly placed spines. So and they can be quite big. In Australia, in Port Phillip Bay, which is Melbourne, they've reached very, very, very high densities. They can consume basically anything that's on the seafloor, so they really like shellfish and other native species. They can go through boom and bust cycles because they're so dominant. They can basically clean out areas and then they reduce in density. They can eat each other. So yeah, there's videos on YouTube if you type in Northern Pacific Sea Star Australia, and you can kind of see what they get up to over there. So this could again enter via vessel biofouling or ballast water. The juveniles or adults via vessel biofouling, and then the larval stage as ballast water mediated. Their larvae can be in the water column for up to 3 months in ideal conditions. So yeah, they can survive quite a while. They can live in the low intertidal to quite deep water, up to 200 meters. They really like a bit like Kalupa, um, sheltered bays and harbors, where that larvae can remain entrained in those water bodies, and that's how they get up to those high densities. They don't seem to get up to high densities in the open ocean, in the open coastline outside of those enclosed harbors. So the impacts will be felt in those more enclosed environments. Moving on to the European shore crab. So page 19 of the Pest ID guide. This is quite a small crab. It's only up to 8 centimeters wide. We've got a picture and a few slides showing the densities they can get up to in an invaded range, and it's pretty outrageous. So these guys are really small. So their key diagnostic features, again, with a lot of crabs, is the lobes or teeth between the eyes. So there's 3 of them there, and then there's 5 spines on each side. And again, it has no swimming paddles. So you put those 3 features together. If you see them, then you're probably onto something, and it's time to give us a call. Again, vessel biofouling and ballast water are the main culprits for entry. Surprise, surprise. And again, high densities, very aggressive, and just eats a lot. Once they get to those high densities, that population can consume a lot of biomass to sustain itself. This is the Asian clam. So this is different to the freshwater gold clam that you may know about. So this is a very small clam. It's only 3 centimeters long. The key feature for this species is that one side of the shell is larger than the other, so it forms an overbite. And that is the key. So very small, and it has this overbite. So this species is present in San Francisco Bay, where it was introduced a number of decades ago, and it reaches vast densities of up to, what does it say there, 25,000 per square meter. So it's a filter feeder, so it's filtering a lot of larvae and organisms out of the water column, and it's the introduction of the species in San Francisco Bay was correlated with a collapse in some of the fishery species in that bay. So it probably contributed to the decline of those fisheries. Again, it likes enclosed areas. It's subtidal, but it can be found between the high and low tide mark too. It likes soft sediments that it can burrow into, and it's pretty tolerant to fluctuations in salinity, which is pretty useful if you're an organism living in an estuarine environment. All right, so they're the species that aren't known to be present in New Zealand at the moment. And so for the rest of these species, they are established in New Zealand, but in localized areas. So let's get to it. So first off is exotic Kalupa. So this is present in the Upper North Island on the east coast, from the Bay of Islands down to the Hauraki Gulf, and the tip of the Coromandel Peninsula. Exotic Clerpa, we call it exotic Clerpa because it's actually two species. They look exactly the same, so you can't look at exotic Clerpa and say whether it's one species or the other. You have to use molecular testing to determine. So for all intents and purposes, it's yeah, just think of it as one one seaweed. It's also bright green, a bit like Clerpa taxifolia, which we talked about before. The key difference is that the leaflet is solid, so it's oar shaped. It's kind of like a blade of grass. It doesn't have the midrib and it doesn't have the pinnules growing out of from that midrib. So it's yeah, it's solid in shape. But it also it's similar size. The fronds are similar size to taxifolia. And it's connected to a horizontal runner, the stolon. So, as a lot of you will know, probably been involved with exotic caulerpa or know about it, read about it, it can reach very high densities, and that has been observed in New Zealand, reaching very high densities. Its habitat, it can grow on a variety of benthic habitats in the marine environment, from sand to mud to reef. It can grow in seagrass beds. It's quite variable in the habitat that it can grow on. Um, yeah, it's been found from, low tide or even a little bit higher than low tide up to 50 meters water depth. Um, there's been a marked decline in its abundance lately within the last year or so. Um, there's been a die-off. No one's really sure why there has been that die-off. It could be a variety of reasons. And it's quite interesting that the die-off has been observed in a number of locations. Um, so that makes it, yeah, even more interesting to try and untangle the cause and effect of that. And the short answer is we're not really sure at the moment. There's been a bit of research done looking at environmental factors over the last few years and trying to see if there's been any key changes and and nothing stood out to correlate with the timing of the die-offs. So yeah, it's a bit of an unknown. Um, Overseas, that's true too. There's been a Caulerpa species spread to other countries where it's not a native species, and there's been that same um, observation of the boom and bust cycles, and then that's it's not unusual for non-indigenous species that go to that establish in new locations to have that boom and bust cycle. Um, and it's always difficult from a scientific perspective to discern what is causing that. So this species most likely entered into New Zealand via entanglement, maybe an anchor or an anchor chain, something like that, some sort of equipment that was that interacted with a better caulerpa overseas. All right, the Asian paddle crab. So this species is quite widespread. The upper northeast coast of New Zealand, from Ohiwa Harbour is about as far south as it's been found so far on the east coast. It's also been found in Raglan Harbour on the west coast. So it's slowly spreading. Um, this is quite a larger um, crab. It's quite aggressive compared to our native crab species. It's a lot more aggressive. So you can almost use that as a diagnostic feature. If it stands its ground, it's probably something to investigate a bit further. So morphologically, it has six spines on each side of its shell, and then it also has spines on the upper surface of each claw. So that's quite a good diagnostic characteristic. Um, it's variable color. Um, but it does have paddles on the back like our native paddle crab. So yeah, those spines are the way to go. It most likely entered New Zealand via vessel biofouling. Um, Again, like a lot of the species we've talked about, it likes estuaries and harbours, um, and sand and muddy habitats. Moving on to the Mediterranean fanworm. So this species was first detected in 2008 in Littleton, and then subsequently about a year later in the Waitemata Harbour. This is definitely entered via vessel biofouling. It's a prolific fouler of man-made structures in the in the marine environment. So yeah, piles and aquaculture equipment, etc. Boats can get to high densities. So the key diagnostic feature here is it has a single crown um, that is used to catch larvae in the water column. So that's what that crown's used for. It's a single crown, spiral. It's got a spiral shape. A lot of native and other fanworm species have multiple crowns out of the same tube, whereas this one only has one. Um, it can be quite large. Yeah, can grow up to the tube can be up to a meter long. Yeah, and they can form very dense aggregations. Can be up to 1,000 individuals per meter squared. Um, and yeah, I'm a bit like a broken record here. At like sheltered locations without too much wave action or, to survive. The club tunicate, this is Styloclava. Um, this species is quite widespread around New Zealand. Um, as its name goes, it's got two little siphons at the end that look like clubs. Um, it's got quite a warty skin and yeah, lumpy skin, leathery skin. Um, the attachment to hard substrate and the stalk is normally longer than its body. So that probably helps it extend out into the water column and get, so it can filter efficiently. Um, it's a prolific fouler of man-made structures, so it can be a nuisance species for vessels, for structures in the marine environment, aquaculture structures, etc. Highly efficient filter feeder, and yeah, it can grow on other organisms too. And this one is definitely a candidate for being introduced via biofouling into the country. And here we have Undaria. So this is kind of like the original non-Indigenous species, first detected in Wellington in 1987. It's spread yeah, to numerous places around the country now, predominantly by vessel biofouling. Um, it has a midrib that you can see there, number one, that goes up the length of the plant, and then you have these blades, these smooth, thin blades that grow out of each side of that blade. The blade here, the big top part of the plant, can die off or erode when water temperatures increase. And so sometimes all you can see is the reproductive structure, which is number 3 there, which is at the base of the plant. Looks like a frilly lasagna. And sometimes only that portion of the plant remains, and then it regrows when the water temperature is sufficient to allow that to occur. So that lasagna is always there. So that is a key diagnostic feature. Um, it can yeah, its habitat requirements are quite broad for Undaria. It does very well in exposed exposed coastal locations and in sheltered areas. Um, yeah, it can deal well with polluted or modified marine environments and can form very dense colonies. Um, yeah, and as you can see from that boat that's been lifted out there, could be a real pain for vessel biofouling management. And this is the lucky last here, Eudostoma elongatum. So this is the Australian droplet tunicate. This is, as you can see, a sausage-shaped white or cream-colored organism. It's actually a colony of organisms, so that sausage is thousands, if not millions, of individual organisms all grouped together. The diameter of the colonies is 5 to 20 centimeters, and the tubes, the sausages can reach up to 1.5 meters in length in optimal conditions, but generally a lot shorter than that. Sometimes there's orange flecks in the tubes, which are the reproductive structures. This species does really well in summer. It can grow on aquaculture equipment, so that's oyster racks there. It can grow on natural rocky reefs that are then exposed at low tide. So you have these white sausages everywhere. Not too good to look at. Um, it does it is affected by freshwater input. So if there's flooding and a big pulse of fresh water, it can reduce in density very significantly until you can almost not find any of it. But then it'll grow back when conditions improve. Yeah, and again, this is a biofouling species, so it's spread around on equipment and boats. All right, so that's the quick rundown of the species. You guys might not remember all of that stuff, but you have the link to the Pest ID guide, so you can always refer back um, if you need to in the future. So I just want to quickly talk about what do you do when you find something unusual, whether that's in the marine environment, freshwater environment, or in the terrestrial environment when you're gardening or walking the dog or whatever. So if you see something unusual, that's you guys all know your specific areas of the country. What do you do? And it won't come as a surprise to many of you that we have a pest and disease hotline. Um, and you can call it to notify us of what you think you've found. So depending on the organism, um, we might need a sample. A photo might be good enough, and you can always ring us if you want, and we can help you take a photo that will allow a positive ID to occur. In some circumstances, we may need a physical sample to be sent to a laboratory to get ID'd, and obviously knowing where the organism was from is vitally important. So we have the pest and disease hotline, so the phone number, but we also have an online notification system. So you can use that too. If it's deemed a high risk notification, you will be referred back to the pest and disease hotline. So yeah, just bear that in mind. So this is kind of the workflow. So we have for notifications. So we have at the top here, we have what we call surveillance. So we have our targeted surveillance programs that central and local government and others run. And then we have our general surveillance, which is public notifications. So we have a huge range of notifiers from a variety of different areas of the community. And then so you'll ring the 0800 hotline. It then gets triaged at our call center, and will then get directed to the relevant team. So the team I showed you at the start that I'm a part of is the aquatics team, but we have other teams for plants and insects, for animal diseases, etc. So the notification gets triaged to the relevant group, and then the on-call incursion investigator will take up that notification and figure out what they need to do. So yeah, the incursion investigators are on call every day of the year during those hours, and we have a key performance KPI that we have to respond within 30 minutes to that notification. So that's kind of how it works, just briefly. This is the picture here of the Chinese midden crab, which has invaded the UK. You can see that is all Chinese midden crabs. So they're really little. So you can see the densities they can get up to, which are, yeah, pretty high. I just wanted to show you guys this little schematic here of what we're trying to do with early detection. So on the y-axis, the bottom axis here, we have time since establishment. So at the left-hand side, that's time 0, and then we move across as we move to the right, and that's a time period. It could be years, months, days. And then on the axis, the y-axis here, cost to manage, you have from 0 cost to increasing cost. And then this red line here is, think of it as an organism being established in New Zealand. So what we're trying to do with early detection and surveillance is detect that organism as close to time 0 from when it established when it came to New Zealand, as we're trying to push it down so we find it as early as possible, and then therefore the cost to manage is reduced. If we find it later, somewhere up higher on this curve, it might have been here for days, months, years. It may be in multiple locations. It may be at very high densities, so therefore the cost to manage is much, much higher. But if we can push it down to the left here, we have better chances of successful management, whether that's eradication or population suppression, etc. So that's kind of what we're all about here and what we're trying to do. And this is where you guys can all be a big help. So getting specimens that are in a condition that allows either an ID to be made for a pest or diagnostic tests to be conducted if you're concerned about a mass mortality event or a specific pathogen is really important. And ringing the 0800 number, the incursion investigator can help you with the exact methodology to do. But it's all about, maybe it's putting things in the fridge over the weekend so you can send it off to the lab on Monday so it's still, either alive or freshly or still fresh, and doing that in a way that preserves the organism's integrity as it's being transported. So yeah, we won't get too much into detail here, but there is a whole process around that the incursion investigator can help you out with. And this slide is just a flow on from that. A good descriptor I heard is that if you want to have high quality samples submitted to a lab for disease testing, it needs to be good enough to eat. If it's rotting or decomposing, you won't be able to conduct the required tests because there'll be secondary bacterial infection confounding those results. So yeah, timely reporting and high specimen quality is key. But we can help you with that. That's not a problem at all. You can just ring the 0800 number and yeah, we can give you the best practice instructions. And then what to expect when submitting samples, either for a pest ID or for disease diagnostic testing. Testing sometimes can take quite a while. For pathogen testing, sometimes you have to run a number of different tests and then you have to interpret those results, and that can take quite a lot of time. And that can be true for pest IDs too. Sometimes you have to do a morphological assessment, and then that has to be paired with genetic testing, and then you have to interpret those results, and that can be that can be a bit of a process. But the incursion investigator will stay in touch and update you as that progresses. And then all the results, they are communicated for pests either on the Marine Biosecurity Porthole or in the surveillance magazine. So the surveillance magazine is published every 3 months and is a really key publication to show overseas markets that we don't have pests and diseases that they'd be concerned about, and it helps us and it supports market access to those overseas markets. On the Marine Biosecurity Porthole, feel free to go on there and have a play. We have a relatively new map that shows if you're interested in a specific location, you can go to this marine pest by location map, and it's very easy to use, and you can get a list of species that have been detected from those areas. So you have the scientific name and the common name there. And finally, we have these resources available if you want them. So you've got links to the Marine Pest ID Guide in the middle here, and a link to the Freshwater Pest Watch List. This is a relatively new publication, is only a few months old. And we also have these cheat sheets on the right here that we can send out. So these are little A4 publications that kind of a cheat sheet, little standard operating procedure about what you should do if you find a aquatic pest or a aquatic disease. So if you would like some of them sent out to you, please just email me after this, and I can facilitate that. Likewise for the Marine Pest ID Guide, I can do that. For these other publications, the Aquatic Disease Guide and the Dive Book, they currently only exist in physical copies, and we have limited numbers of them. So, yeah, if you want them, let me know, and we can see what we can do. Awesome, thank you. That is the conclusion of the talk.
Thank you very much, Abe. Very, very interesting and great to have some really excellent new material in there too. There were a couple of questions that came up in the chat box and the Q&A. So one question relates to, is there any research or modelling being conducted around the link around how climate change and increasing sea temperature might affect the distribution of marine pests?
Yeah, there is some literature, for sure, like the marine heat waves that have been happening the last few years up the especially on the northeast coast of the North Island. There is uh, I'll try and find the publication. It's around the Ascidian Symplegmia brachyhyme, I think that's the name, and how the heat waves correlated and increased density and distribution of that species. So definitely a lot of these species can do well when water's warm, and that obviously increases the establishment likelihoods for a lot of these species that like warmer waters. So yes, definitely a longer term and shorter term issue that we're grappling with, and hence why the preventative measures are so important, right? If we can stop things getting here in the first instance, then you know, the habitat and the habitat suitability doesn't matter so much. But yeah, for things that do get here, that can, depending on the species, of course, increase the likelihood of establishment.
Thank you. Do you have any insight, this is again in response to a question, into how that might affect the possible spread of callirpa to the South Island?
Yeah, that's another good question. Yeah, it'll depend because it's not just that it'll be with callirpa because it's got boom and bust even up in the upper North Island where the water temperature is much warmer. Um, there might be some sort of threshold for where it can establish and not, and it might be, say, in the summer months it potentially could establish, potentially, I'm not sure, depending on what the water temperature is at your location. But there might be thresholds where the winter temperatures, you know, it can't do well because it's too cold, and therefore it dies off, and yeah, it's not suitable. But with climate change, maybe over what time frames, I'm not sure. Um, there's quite a bit of work looking into um, habitat, what the thresholds are for temperature and light and other environmental parameters for Kalupa establishment. So hopefully this research will help inform that in a bit more of a data-driven way. Yeah, it's always hard to know because these organisms, you see how they act overseas, and I think I've said this before, and many of you will know this, but they can act differently when they come to a new environment, and the kind of rule book gets chucked out for a bit for some reasons that we don't know, right? Like it might be certain pathogens not here, or yeah, not sure. They keep us on our toes.
Now, how would you respond to someone who said marine pests aren't an issue because we can eat them?
It would depend on the marine pest. Yeah, it's it's all about the values, right? It's what values do you hold dear? And there's economic ones, there's socio-cultural values, there's economic values, and values can change over time. Um, so Asian paddle crab, it would depend where it is. If it's in a fully established place where it's technically not feasible to get rid of it, then it's, you know, you kind of have to learn to live with it because at this stage you can't get rid of it. Here's the story. Oh, I haven't seen that. Um, yeah, you have to make sure you're allowed to eat it too. Especially looking at some councils manage the Asian paddle crab's part of the pest management plan of some councils. So you just have to check on the rules there. Um, but yeah, if that's allowed, then hey, eating it's yeah, fine.
And there's only one question left so far, unless anyone has anything to add. Is there a cell phone app to help us identify pests and engage with Biosecurity New Zealand? Can can the pests be identified using the Aotearoa species identifier app?
Oh, I don't know. That's a good question. I'll have a look at that and yeah, send me an email and I can yeah, I'll have a look. Yeah, I don't know how good that the app is, but I know that those, but like iNaturalist and that their automated IDs are getting better and better with from photographs. But it'll depend on how good the photograph is and what the diagnostic features are of the species and how they can be discerned from native species. So there's always a little bit of yeah, a few steps to work through there. So yeah, sorry, I can't give you an answer.
And another question, are there any consequences for vessels bringing these species to New Zealand on their hulls?
Yeah, well, we have a craft risk management standard for vessel biofouling, and also there's rules for ballast water. So if those vessels are dirty, then yeah, they can be delayed in their itineraries, and that costs them. There's no fines per se, but messing with a commercial vessel's itinerary can cost them tens, if not hundreds of thousands of dollars a day. So they want to avoid that at all possible. Yeah, they want to avoid that delay. So that is a pretty big incentive for them to comply with that standard.
And even for recreational vessels, that
Yeah. Yep.
yeah, same thing
Yep.
applies.
Yep.
They're required to be cleaned, and that's expensive. It's a cost that they incur. It's time in their time delays in their travel, etc. Tess has asked, is iNaturalist actively monitored for invasive species postings? We know it's
Yeah. No,
successful at times, but
that's a great question, Tess. Yes, yes, it is. That's how exotic caliper was first detected. was a photograph on a posting on iNaturalist. So yeah, it's a very good source of data.
Right. I think we've gone through all the questions. Thank you very much, Abe, for your time and knowledge. I should have mentioned at the beginning that this webinar is organised with Abe's input by the top of the North Marine Biosecurity Group and the top of the South Marine Biosecurity Partnership, who organised the Clean Below Good to Go campaign. So there's a whole lot of work around education and engagement with boat owners to help prevent the spread of marine pests involved in that. And the website for that is marinepests.nz. Yeah. Thank you. We'll sign out now.
No worries. Hey, thanks, team.
Thank you.